Spica Therapeutics, a stealthy biotech based in Antwerp and Aarhus, aims to leapfrog existing cancer and fibrosis treatments by precisely targeting pathogenic immune cells called macrophages. Hey, welcome back! Before we dive into this biotech startup, consider subscribing for more healthcare innovation insights and company deep dives. Let's begin with the basics. What does Spica do? What problem is it solving? And how big is that opportunity? In short, Spica Therapeutics is a preclinical biotech startup advancing a class of anti-𝗠𝗔𝗏 monoclonal antibodies. These unique antibodies are engineered to selectively deplete pathogenic macrophage subsets, especially those expressing the 𝗠𝗔𝗏 antigen. To understand this, let's break down what macrophages are and why they are such a compelling therapeutic target. Macrophages are specialized white blood cells that play a central role in our immune system. Think of them as the front line of defense against infections and injuries. When activated, these cells engulf and destroy pathogens, cellular debris, and even cancer cells. However, not all macrophages are created equal. Different subsets can have distinct functions and sometimes even conflicting roles in the body. In cancer, for instance, certain macrophage subsets promote tumor growth, immune evasion, and spread, while other subsets help activate anti-tumor T cells and directly kill cancer cells. So it's not about eliminating all macrophages, but rather selectively targeting the harmful ones while preserving the beneficial subsets. That's where Spica's lead product candidate comes in. Its depleting anti-𝗠𝗔𝗏 monoclonal antibodies are designed to target and remove pathogenic disease-driving macrophages that express the 𝗠𝗔 protein. This approach is particularly promising in two areas: cancer progression and fibroinflammatory diseases. In cancer, tumor-associated macrophages TAMs often skew the immune microenvironment to favor tumor growth and spread. By removing these pathogenic TAMs, Spica aims to restore the balance of the immune system to fight the cancer more effectively. For example, in breast cancer, the abundance of TAMs is associated with lymph node metastasis and poor prognosis. Interestingly, roughly 50% of TAMs express 𝗠𝗏, making it a good target for Spica's therapy. In fibroinflammatory diseases like idiopathic pulmonary fibrosis (IPF), unwanted scarring or fibrosis is driven by specific macrophage subsets. By eliminating these cells with its anti-𝗠𝗏 antibody, Spica aims to limit the progression of fibrosis and alleviate symptoms. So why is this approach promising, and how might it fit into the bigger picture of cancer treatment and fibrosis management? In cancer, most current treatments like chemotherapy, radiation, and surgery are nonspecific, affecting both healthy cells and the immune system. While checkpoint inhibitors have revolutionized some cancers, a significant portion of tumors remain resistant to these therapies. Additionally, not all patients respond, and many face immune-related side effects. Spica's targeted approach offers a way to remove the tumor-promoting immune cells, potentially enhancing the efficacy of existing treatments or making it easier for the immune system to recognize and attack the cancer. In IPF and other fibroinflammatory diseases, current treatments mainly focus on slowing the progression of fibrosis. There is still a substantial unmet need for effective disease-modifying therapies. Spica's approach could provide a much-needed solution by directly targeting the disease-driving immune cells. By selectively removing harmful macrophages, Spica's anti-𝗠𝗏 antibody has the potential to avoid the limitations of current therapies and provide a safer, more targeted treatment option. The core value proposition revolves around three key aspects: better clinical outcomes, improved patient experience, and real-world evidence. In terms of clinical benefits, Spica anticipates that its targeted macrophage depletion will lead to improved progression-free survival (PFS) and overall survival (OS) in select cancers, especially those with high macrophage infiltration. For example, preclinical studies in the 𝗠𝗔𝗏𝗥𝗢𝗣𝗛 breast cancer model show striking regressions when combining Spica's anti-CD163 antibody with chemotherapy. Notably, this combination reduced lung metastasis by over 90% compared to chemotherapy alone. Beyond survival, the antibody is expected to enhance patient quality of life by reducing treatment-related toxicities and immune-related adverse events, which are common with checkpoint inhibitors. This aspect is crucial because it can lead to increased patient adherence to treatment and better persistence. From a real-world evidence standpoint, the targeted approach should make it easier to track the effects of the therapy using imaging techniques like positron emission tomography (PET) and magnetic resonance imaging (MRI). For example, CD163 PET imaging could help identify patients who are more likely to benefit from the therapy upfront, enabling biomarker-guided precision medicine. All of these improvements position Spica Therapeutics to address a significant unmet need in the market, offering better outcomes for patients while also providing payers with a more cost-effective treatment option. The value drivers related to better clinical outcomes, improved quality of life, and real-world evidence support a strong market opportunity for Spica Therapeutics. Now, before diving into competitive benchmarking and valuation, let's talk about the technology push and market pull that makes Spica's emergence particularly timely. The technological push aspect refers to recent advances in our understanding of macrophages and the tools to study them. Breakthroughs in fields like single-cell transcriptomics and spatial immunophenotyping have unveiled the complexity and heterogeneity of macrophages. We now know that different macrophage subsets play diverse roles in health and disease. This knowledge has paved the way for new therapies aimed at selectively targeting specific macrophage subsets involved in disease progression. Spatial phenotyping tools like multiplex immunofluorescence and mass cytometry have enabled researchers to visualize and analyze the tumor microenvironment at a single-cell level. This has helped identify which macrophage subsets are most closely linked to tumor progression and predict patient responses to therapy. Advances in imaging techniques like PET and MRI have also made it possible to monitor the accumulation of macrophages in various diseases, opening doors for non-invasive diagnosis and treatment monitoring. On the market pull side, there remains a significant unmet need in several disease areas where specific macrophage subsets play a detrimental role. In oncology, while checkpoint inhibitors have transformed treatment landscapes, challenges remain. About 60% of tumors do not have high enough PD-L1 expression to benefit from current checkpoint blockade, and those that do respond often face issues with acquired resistance and immune-related side effects. Moreover, the cost of checkpoint inhibitors is substantial, reaching approximately $150,000 per year in the US. Spica's approach could address some of these challenges by selectively removing the tumor-promoting immune cells, potentially making existing therapies more effective or reducing the need for costly checkpoint inhibitors in suitable patient populations. Similarly, in fibroinflammatory diseases like idiopathic pulmonary fibrosis (IPF), current treatments only modestly slow down the disease progression and cannot halt it completely. Patients often have a poor prognosis with a median survival of 2 to 5 years after diagnosis. Spica's disease-modifying therapy could provide much-needed improvement in these areas. The potential to address significant unmet needs in both cancer and fibrosis create a compelling market opportunity for Spica Therapeutics. Moving on to the competitive benchmarking, Spica Therapeutics is part of a small but growing field of innovators focused on harnessing the power of macrophages for therapeutic benefit. However, it is worth noting that none of these companies operate in exactly the same space. One notable competitor is Carisma Therapeutics, which is developing chimeric antigen receptor macrophages (car-macrophages) for cancer immunotherapy. The company's lead candidate, CM24, is designed to target CD19 positive cells. While this approach holds promise, it involves higher complexity, greater cost, and more challenging logistics for manufacturing and delivery compared to Spica's depleting antibody platform. 𝗧𝗏 is currently in early-phase clinical trials, so it faces significant hurdles to overcome before its therapy becomes available to patients. Another competitor is Macrophage Therapeutics, which is focused on broadly activating macrophages to enhance their anti-tumor activity. However, this approach is less selective than Spica and may result in a less predictable efficacy and safety profile. Macrophage Therapeutics is at or near in readiness, meaning it is still in the early stages of development and has not yet progressed to clinical trials. What sets Spica apart is its simple, targeted, and easily scalable strategy compared to the more complex cell therapies. It allows for flexible development and commercialization with the potential for wider applicability across various diseases and patient populations. Spica Therapeutics is pursuing a market access strategy centered around biomarker-guided precision medicine, which should simplify interactions with regulators and payers. Key elements of this strategy include the development of companion diagnostics to identify ideal patient populations seeking orphan or fast-track regulatory designations in oncology and rare fibroinflammatory diseases, and generating early health economics and outcomes research models to quantify its value. This includes demonstrating reductions in hospitalizations, improvements in quality-adjusted life years (QALEs), and shorter time to remission or other clinically meaningful endpoints. By focusing on clear clinical and economic benefits and tailoring the therapy to specific patient subsets, Spica aims to position itself for payer acceptance and reimbursement negotiations. Regarding valuation, as a privately held company, Spica Therapeutics does not have publicly disclosed financial information. However, recent news of a €10 million seed round likely values the company between €30 and €50 million pre-money. Comparable private companies working on macrophage-targeting or tumor microenvironment therapies at a similar stage have reached Series A or Series B valuations between €60 million and €100 million. This is usually dependent on data maturity and platform extensibility. Assuming a risk-adjusted peak revenue potential of €500 million and a typical discount rate of 25% reflecting early stage biotech risk, the company's long-term risk-adjusted valuation could land in the range of €150 million to €300 million over a 5-year timeline. This implies a potential 5 to 10 times return on the seed investment if key milestones are achieved. Overall, Spica Therapeutics is well-positioned to capitalize on recent advances in macrophage biology and the growing need for targeted therapies in oncology and fibrosis. Its unique platform, biomarker-driven approach, and focus on real-world evidence support a compelling market opportunity. With execution on early milestones and clinical validation, Spica could become an attractive acquisition or IPO candidate within the next 4 to 5 years. That's a look at Spica Therapeutics. Do you see opportunities in macrophage-focused therapies? Comment below. Thanks for watching. If you enjoyed this video, please hit the like button. Until next time.